In this video, I want to describe the different types of mutations, and specifically single point mutations, that can occur and then their ultimate change in the protein. So when we talk about mutations and DNA mutations, we need to remember and go back to the central dogma of biology. And that's the concept that this molecule DNA that resides in the nucleus is used as a template for an RNA molecule. Ultimately, this RNA molecule in the form of a messenger RNA is read by a ribosome for the ultimate production of a protein. So in terms of mutations, we are going to kind of talk about how a mutation at the level of the DNA may ultimately lead to a change in a sequence of amino acids or proteins. Okay, so the first class I want to talk about is what we call single point mutations. So these are really looking at a single base pair change. So we're only going to change one nucleotide in the DNA and then ultimately see how that change is seen at the level of a protein. All right? And so what I'm going to do, just for simplicity's sake, is pick a codon, a triplet codon, and I'm going to give you the triplet codon in RNA form. I will then briefly show you how we can convert that back to DNA, but for simplicity's sake, everything's going to be in RNA. And the codon I'm going to choose is UUA. So if we translate this into a protein or an amino acid, this will encode for the amino acid leucine, or LUU. All right. So now I'm going to go through a series of mutations to this codon so we can ultimately see how it changes or doesn't change the amino acid. Okay, the first change I want to do to this codon is actually going from UUA to CUA. So as you can see, I'm changing this position. This is where the mutation is occurring. And this leads to the amino acid leucine again. So because of the redundant nature of the codon table, you can see that we have made a mutation at the level of RNA here, and I'll show you in a second the DNA, and it leads to ultimately no mutation at the level of the amino acid. We refer to this as a silent mutation. So we're changing the DNA, the nucleic acid, but there's no change at the level of the protein. If we then go from UUA and change it to UAA, we're going to go from a leucine actually now to a stop codon. So now actually from encoding an amino acid in the first place, we now are going to stop the production of the protein at the ribosome. This is referred to as a nonsense mutation. If I go from UUA to UUU, we will go from our leucine now to a phenylalanine. Because we changed the amino acid, this will be referred to as a missense mutation. So once again, a nonsense is moving from an amino acid to an actual stop codon. And in this case, going from UUA and mutating this third position, I am going from UUA to UUU, we're going to incorporate a phenylalanine, and we're changing the amino acid from leucine to a missense mutation. Okay, now if we go a step further and go from UAA again to AUA, so we're changing this first position, this will lead to isoleucine. This is indeed a change, so this is a missense mutation, but it's not a very drastic one in terms of the chemistry of it. So in terms of this amino acid, its chemistry is fairly similar to that of leucine. So this is still a missense mutation, but we can more define it as a neutral mutation, because chemically it's not changing a whole lot, so it may not lead to a huge change at the level of the protein. But in some cases it indeed may. So once again, if we go back, we've got the concept that in this case, when we change the first letter from a U to a C, so we've mutated it, we have a silent mutation because there's ultimately no change in the amino acid. If we change this second position from a U to an A, we will incorporate now a stop codon, so we refer to this as a nonsense mutation. And from this third position, we change from an A to a U, we're going from leucine to phenylalanine. Because we changed the amino acid, we refer to this as a missense mutation. And then UUA to AUA, changes first position to an isoleucine. It's a missense, but more defined as a neutral because chemically it's not a huge change. So just to show you this UUA in terms of nucleic acid, if we know that our mRNA codon is UUA, one strand of our DNA will be complementary to that, so that would be our template, and that's going to be AAU, and so then the other strand will be TTA. All right. So once again, DNA is double-stranded, RNA is single-stranded, and then so if we take a change at this level, that change will be perpetuated down the line to the mRNA, and then ultimately, as we saw over here, we can see the changes in the amino acids, or in some case, because of the redundancy of the code, we will see no change in the amino acid that's produced.